In this video we will discuss about FAK tail binding is necessary for wound closure or not. In order to check uh, this question, what they did is they developed in vitro models. From the in vitro 3D models to check the wound closures of the tissues, in which they use different types of FAK. That is, in one condition they use a wild type FAK, in another condition they use the mutated FAK where tail is absent or tail is mutated using this E1015 with A, and in one case they do not use FAK. And this is in the panel A. They shows their model where what it is at time 0 they seed their model with this collagen gel, and after 24 hours they allow this macro tissue formations. So at time 0 they seed the cells along with this collagen gels, and after 24 hours they see the macro tissue formations, and then they make a wound and check the wound closures after every 1 hour, and after 48 hours they check after 48 hours means the 48 hours from this seeding of the collagens and 24 hours from this wound has been created. They check the evaluation of these activations of the phosphorylation using PY397 phosphorylation at Y397, and also check the wound closure rates. So the results are shown in this panel B, where we can see that in the panel B, when the wound is created, initially the wound area increased, as we can see in the wound area, the raw data and also the normalized data. Wound area first increased and then it decreased slowly with time. But for this E1015A, or where the tail is mutated, we can see that FAK is mutated. This is the appropriate one where the FAK is mutated, and we can see that in this case the wound closures occurs but at a slower rate, and in some conditions they do not see any wound closures, and in the null case where they do not have any FAK, they do not see any wound closures. That can be seen here. And thereafter they also checked some other parameters such as amplitude, which can show how much stress the cells has gone through. So for the wild types they gone through a maximum amount of stress compared to the mutated FAK, and compared to when there is no FAKs are present. And the wound closure rates are also follows the same trend. That is, wound closure rates are higher for the wild type than E1015A or the mutated FAK, and for the null they do not see any wound closures, or the wound closure rates are minimum. And the force, thereafter they check the force also, change per post, and the force that exerted on the macro tissues seems to be maximum in the wild type compared to the mutated, and also compared to the case where no FAKs are there. And when they check the width contractions at the center of the wound closures, what they are able to find out that the tissue with contractions for the wild type and also for this mutated FAK is higher than the case where no FAKs are used. And when they check the activations of this FAK integrated intensity, they see that it is maximum for the wild types, then comes the mutated FAK, and for the null they see a minimum activations of the phosphorylations because they do not have the FAKs. So this you can consider as a background signals. And in the panel H they have shown the image or immunostaining image of the different parameters. That is, for the DNA we can see that, and for the TFAKs you can see the intensities are higher for the wild type, then it goes down for the mutated, and it is minimum for the case where no FAKs are there. And the activations also follows the same trend, and similarly the effectins also follows the same trend. And for the control, they use IgG, and IgG's intensity seems similar, indicating that their staining does not get affected, and they do not have any they do not have any uh, batch effect or human errors while measuring this immunostaining images. And then they check the heat map also, and in this case they can see that for the DNA it seems intensity seems similar, but for the TFAK we can clearly see that intensities are higher for the wild type than its minimum for the E1015A, and lowest or negligible for the case where FAKs are not present. And the activations of the phosphorylations also follows the same trends, highest for the wild type, then comes the mutated FAK, and then comes the null or no FAKs. And the effectins is also follow the same trends. It is highest for the wild type, then comes the mutated FAK, and then the lowest one does not have, lowest one has the minimum intensities, or lowest one, or the where there is no FAKs. And for the control, we can see the IgG control intensities looks similar. So these all immunostaining results also in line with the quantitative analysis that are shown in this figure. So this in overall indicates that FAK tail bindings are necessary for the wound closures, and it will have a highest wound rate compared to the mutated one. And without FAK, they do not see any wound closures. If you need to understand more about the experiments in detail and their in vitro models also in detail, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.